So last speaker we have Nipam Patel, colors in butterflies.
So I'd like to thank the organizers again for the invitation to this meeting, and I've enjoyed listening to all the talks. And so one of the things that my lab's worked on for a number of years now is trying to understand coloration in butterflies, but a pretty specific example of color. So obviously butterflies are a nice charismatic group of organisms that probably everyone recognizes and appreciates the colors and patterns in. And a lot of researchers and a lot of success now has sort of gone forward in understanding the patterns that you see on the wings of butterflies. And a lot of that comes from genetic approaches and approaches that use interbreeding between heliconius butterflies that have different forms. But what I'm going to focus on is how the butterflies actually get the color, and very specific colors, namely greens and blues in particular. So if you've ever touched the wing of a butterfly, right, your mom always told you not to touch the wings of the butterflies because they couldn't fly anymore. So it does damage them because what you're doing is you're pulling off these things which are scales. And so the scales are a dead remnant of a cell that had been growing during the pupal stage. And so it's an extension of a cell. So for a cell then it's pretty massive. So a typical scale is about 300 microns long and about 50 microns wide. The cell body sits down in the pupal wing as it's growing, and this is an extension that comes out.
And fairly squamous? Sorry, very thin?
They're flattened, yeah. They become flattened. You'll see pictures in a minute. Towards the end of pupal development, or actually after the butterfly closes, the cells are actually, these aren't alive anymore. Okay? But before they die, they can deposit, in this case what you're seeing are pigment molecules that have been put into the scale, which gives it color. And so pigments are responsible for a lot of the colors like whites and browns and yellows and things like that. Okay, so it turns out that that part of the spectrum, so the blacks, browns, yellows, oranges, are all coming usually, or almost always, from pigments in the scales. And so what do we mean by pigments? So pigments are going to be molecules that have particular absorption properties. And a good illustration comes from thinking about what's in highlighters to give them color. So these are individual compounds, and in this case we often use cyclic carbon compounds, that absorb specific wavelengths of light. And by doing that, the light that's left is reflected, and that gives you the perception of color. So for example, your yellow highlighter has a molecule in it that is very good at absorbing blue light, and so then the remaining light that's left is what gives you the perception of yellow light. So that's how pigmentary colors work. But the colors I'm going to focus on today are what are called structural colors that are made by structural mechanisms instead. And in butterflies, these are largely relegated to short wavelengths. So I'll show you examples where you can make every color that you want structurally. But in many cases in butterflies, the blues and greens that you see are not made by pigment molecules. So these short wavelength colors are instead made as what we call structural color. And I'll explain that in a minute, but I want to just add that this isn't unique to butterflies. So there are plenty of examples of structural color throughout nature. So many of the iridescent blues and greens that you see in feathers are also made this way. Even the blue skin of monkeys is actually a structural color, so it's not created by pigment. It's actually created by an underlying network of keratin at the right distance under the skin. Blue eyes in humans, and things like the iridophores, the shiny colors that you see in fish, are all examples of structural color. So, yeah.
There are examples of blue pigment colors, though?
Yes, there are. Blue and green pigment colors. They definitely exist, right? So, and we can come back to the question of why do you pick one solution over another. And there may be no answer to the why. It may just be it works, and whatever happens is fine. Okay, so how does structural color work? So it works in the same way that when you see color in a soap bubble. Okay, so the soap doesn't have any color to it, right? So why does the bubble have all of these colors of light? And this comes down to a simple sort of physical property that people had found, more than 100 years ago and began to understand. So the first thing that happens is when light hits a surface of a different refractive index, right? So in this case, you're seeing this beam of light hitting this piece of glass, actually. And so one thing that happens is that you get a reflection in each surface when there's a change in refractive index, okay? But interesting things happen when this becomes very thin, okay? So in this case, and chitin is the exoskeleton material that insects are made out of. and it has actually a relatively high refractive index in this air, which is 1. And so when you have this heightened layer, what's going to happen is you're going to get a reflection of the top surface. You're also going to get a reflection of the bottom surface. Okay, and then what's interesting is what happens to these waves as they potentially interact with one another after they've been reflected. And so if those waves are in phase, right, you can get this constructive interference. And so when that happens, the magnitude doubles, the actual apparent intensity of that light is actually increased fourfold. And if they're out of phase, completely out of phase, then you get destructive interference, and then that light appears not to be there, right? And this is going to be different for different colors of light because the wavelength of light is different for different colors, okay? So that means that different wavelengths are actually going to undergo either destructive or destructive interference or some combination of those things. And so again, the math was worked out a very long time ago that if you start to have materials that are thin, and so this is something around half a wavelength of light, then you're going to start getting a very interesting property of color that comes off of that. And what this equation gives you here, and so it's based on the thickness of this and the refractive index of the two materials, and the angle of the incident light. What you're going to get if you solve this, and m is just an integer, so these will give you the wavelengths of light that have maximum constructive interference. And if you remove that 1/2 value, you'll get the wavelengths that have maximum destructive interference. Okay? So in this way, a thin layer of chitin can actually generate color, even though there's no pigment in there. You can intensify this by laying these, or stacking these layers one over the other, and so you can continue to intensify that effect. And here in this next picture is a very good example of, in this case, a moth that actually can create every color of the rainbow in this manner. So in this moth, called the sunset moth, the only pigment in it is black, and all the colors that you see are created structurally. So these scales all have different layerings of chitin in air, and that different thicknesses of the chitin, and that's giving you this rainbow effect of color. But you can see then that the butterfly can generate the entire spectrum of colors in this manner.
Is there a reason for that?
Is there a reason why they have color?
No, just is it benefit to have this kind of structure?
So we'll get back to that to the end when I start explaining some of the structures, and you can see there are some interesting things. So there's an iridescence effect if you do things structurally. So there's some ideas that you can signal. There are a lot of ideas out there, but I think that many times you'll see examples where I think a pigment or a structural color could have gotten you the same solution, right? So it's not necessarily that one is better than the other. Though there are people who will tell you that it's actually hard to have pigment molecules that efficiently absorb long wavelengths of light. So the big exception in the world is chlorophyll, which is why the world is green. Okay, but that's actually a rare molecule that's very efficient at absorbing long wavelengths of light. Okay. So here's another example of structural color that may be familiar to everyone. So these are these blue morpho butterflies, big metallic blue butterflies that you see. And so one easy way to show that this is a structural color is what you're going to see in this movie is we're dropping a drop of acetone on here. And so you see now it suddenly goes green instead of blue. And that's because now instead of going from air to chitin, you're going from acetone, which has a refractive index of 1.3, into chitin. So the equation changes. Right, but the beauty is that this acetone is going to evaporate away, right? It'll go back to just being the normal blue as the air replaces the acetone space, right? So it's gone back to being blue. And so that's one way, very easily in butterflies, to tell that something is a structural color. If I were to put xylene on here, and xylene has the same refractive index as chitin, I would cancel out all structural effects, and then the wing would be brown, which is the normal color of just the chitin itself. Okay, so how do these guys create this structural blue? So if you zoom in on the scales themselves seem to be this brilliant blue color. If you go to EM, you see that this is what a scale looks like, and it has these ridges. And all scales have these kind of ridge structures on them. But in these morpho butterflies, these ridges are incredibly elaborated. So these are those ridges, and if you were to cut a cross-section there and look at it on its side, you would see this. And so you've got this incredible Christmas tree kind of shape. So it turns out that the spacing between the leaves and the spacing between these branches is just right to get this massive constructive interference with blue light and destructive interference with most other wavelengths of light. And so that's what gives you that incredible blue color. Okay, so I pointed out that the other color that's often made structurally in butterflies is green. And so green is an especially useful color because it's a camouflage color. So these little hair streak butterflies, which you would find around here in the springtime, and when they sit in vegetation, they blend in quite well. So there's only 2 genera of butterflies that have ever invented a green pigment, and every other solution is structural. And they've invented green over and over again, and they've come up to lots of really interesting solutions to doing that. These butterflies have a particularly unusual solution. So if you look at an individual scale from that butterfly, we're just shining white light down on it, but what you're seeing is that because of the constructive interference, you're just seeing green light come back at you. And you see that that seems to be in facets inside of the scales as opposed to on the ridges like you see from morphos. If you crack open one of these scales, what you see is that there are these honeycomb structures in here. And these honeycombs have a very specific shape. They're called gyroids. And so this is a very interesting example because this gyroid shape was a theoretical mathematical shape in the 1970s, and it turns out that it exists in the real world inside of the scale of these butterflies. And the geometry is just right at this particular scale, in this size scale, that it creates constructive interference of green light. And so that's how these animals manage to be created. So there are lots of examples, and none of this work is from my lab. So this is all work from optical physicists who are very, very successful. In fact, it seems that every physicist that has access to an SEM or a TEM seems to do these things in published papers, where they're find a butterfly, take a scale off, section it, work out the shapes and work out the math, and tell you exactly why it creates the color that you see. And then a few of them are really resourceful, and they'll spend like $100,000 and they'll make a square millimeter of it using a titanium dioxide and a microwave, and then they seem very happy with themselves, which is great. And then they write papers about how this is going to make currency, uncounterfeitable. But I don't understand if it takes $100,000 to make a millimeter. But anyway, as a developmental biologist, right, what I'm really interested in is how does the butterfly do this, right? So these are amazing, precise structures that have to be made. So if they're 10 nanometers off, it's the wrong color. It's not going to do the butterfly any good. And they seem to be able to do it quite easily. So there's millions of butterflies out there creating structural color all the time. Yeah.
Okay, are there other minimal surfaces, or is it just Chinese?
Oh no, there are so many solutions to the problem, right? So basically, they're only limited by the laws of physics, and you will see just an incredible array of solutions that we can come up with to create greens in particular. Okay, so what we've been doing in my lab is trying to understand in detail, first of all, how you make a scale, right? Because in all of these cases, it's modifying scale development in some way to create the structural components. And then once we understand how you build a scale, how do you modify that scale in order to create structural color? Okay, so this is again an SEM of scales on a butterfly, an adult butterfly wing. And so here's that scale structure with the ridges, and again, they're about 300 microns long, about 50 microns wide. And they're held into the wing by a socket, so another cell that's actually at the base of it holding it in place. So when do these scales grow? So if you dissect out the imaginal discs in the larvae, so if you take the caterpillar, you dissect out those imaginal discs, this is what they look like.
This is just autofluorescence over here.
What's that?
This is just autofluorescence.
It's these things. You're seeing nuclei. Yeah. And so you see these imaginal discs, but at this point there are no scales on here. In fact, the precursor to the scale hasn't even been born yet. So it's a flat epithelia. There's a lot of molecular patterning that's going on. So for example, where these eye spots are going to be is already being determined, but it's just otherwise a smooth epithelia other than the wing veins which are down inside. Scale development doesn't actually occur until the animal pupates. So once it pupates, in most butterflies about 12 hours after pupation, then the events are going to begin where the epithelia is going to start to differentiate. And it's not till the very end of development that you see the pigment colors come in. Okay? But what happens in that interval? So about 12 hours after pupation, you start to see large cells form in nice rows across the wing. And these are what are called the sensory organ precursor cells. And they're going to divide into a pair of cells. And in this case, the larger one is the nucleus that's going to be the scale, and the smaller one is the socket.
We're looking down?
Yeah, you're looking onto the wing surface.
And what's the size of those vacuole?
So this cell is probably about 3 microns across.
And why is it black inside if there's no...
Oh, so sorry, this is just a stain that's stained the cytoplasm. Yeah. Okay, so, all right. So then once those two cells are formed, then the scale cell is actually going to start to grow. Do you have a question?
So they're sister cells, the
Yeah.
socket and the scale.
Yeah, they're sister cells. Well, I'll say that's what's in the literature. Yeah. Okay. So then what you're going to see is that the scale cell is going to start to send a projection out through the plane of the wing. Okay. And what you're seeing here in yellow is filamentous actin. So this is the cytoskeletal component that they're using to grow out the scale. The scale grows very quickly, and so now you're seeing the membrane of those scales, and they're tiling, and they're all pointing towards the edge of the wing. And again, if you look inside, this is the filamentous actin inside, which is forming a regular array of rods inside of there. These are massive rods of actin that are forming.
And it's more or less growing flat at this stage?
No, you'll see a picture here in a second. So here's a cross section through one of these scales, all right? So you can see it's round. It's filled inside of the And so the pink again is the membrane, and the yellow is the filamentous actin. And this is an optical cross section through a couple of these. And what you'll see is the actin is very nicely organized into rods just underneath the membrane of the scale. And then there's some larger rods also that are just inside the surface, but they are on the lower surface of the scale going down into the cell.
And that's now a cross-section of a scale?
Yeah, of a growing scale, so just a little.
And it's growing out of the plane?
Yeah, in this sense, it would be growing towards you. Yeah. Okay. And then if we go a little bit later, now the pink is actually the chitin being laid down, and the yellow again is the filamentous actin. And what you see is that you begin to lay down chitin between the actin rods. So the rods get thicker and thicker and fewer as development goes. And so just for scale now, so that initial bud was about 10 microns across, and at this point it's gotten flattened and it's about 35 microns across.
And what sets the distance between the actin filaments?
So, I mean, it's different depending on the scale you're looking at. The different types of scales or different species.
Is there a scaffold? Is there a membrane scaffold from which the actin is?
So there's a uniform membrane across this, right? And then the actin is arranged in rods underneath that. And now what's happening is that the chitin is being secreted between the actin rods. And that may be relatively passive because the actin obscures any chance for vesicles to get to the surface, so they're forced in between the actin.
But do you have a sense as to why the actin is growing in rods?
No. I mean, there's a lot of interesting information from fly bristles, which are homologous to these things about what organizes it. But these are far, far bigger rods of actin than you see in intracellular bristles. And so we don't know how they get organized this way. We'll get to trying to figure that out.
Yeah, where's the nucleus in
The nucleus is a long ways away down inside the wing epithelia. And so this is a giant projection coming out. All right. So then the next thing that the scale does is it destroys the actin, right? So now it's going to sever the actin and get rid of it. And it does that because now it's got an external skeleton. It's starting to secrete the chitin as the exoskeleton it wants, which is now holding up the scale, and it starts to get rid of the actin and then finishes secreting the chitin. And so sure enough, you can show that this F actin really is important because if you take the scales, right, here that's normally developing, and you add cytoclasin D, which depolymerizes the actin, then the scales completely collapse and fall apart because this is their internal skeleton. Right? If you wait until the chitin starts to get secreted and then you depolymerize the actin, the scale's okay, but that makes sense because the cell was about to break down its actin anyway. It doesn't need it at this point.
Does that include that extra actin that was in the bottom that was inside it?
It all gets cleaved apart. Yeah. The lower one were just some rods that were just internal. And they're interesting because they're the only ones that go all the way back into the cell body.
What's that for?
What's that?
What's the purpose of them?
We don't know. We don't know. We our wild guess is that helps to orient the growth. So all the others are circumferential. Those ones are only on one side. It may orient the extension flat, so they're all parallel to each other, the scales themselves. Right. So why have I told you about how you make this actin cytoskeleton? And it relates to one of the other butterflies that we're studying, which is this butterfly here called the emerald green swallowtail. So it's a butterfly that's native to Southeast Asia, and it has these beautiful green bands on it. And actually, it does sit with its wings open like this, unlike most butterflies which sit with its wings closed. And the green actually is a camouflage color, so it blends into the vegetation that it sits on, the shadows and the vegetation that it sits in, and it sits like this with its wings open. So how does it make green? So if you zoom in on one of its scales, it looks like this. Yeah.
Sorry, just in the previous context, when you were showing us the actin filaments, if you look or dive for actin nucleating proteins along.
We haven't done anything like that. Yeah. But that'll become very relevant in just a minute to understand. Yeah. Okay, so this Palinaris scale looks like this, right? And so what it is it's composed of all of these dimples. And these dimples are actually a multi-layer of chitin in air. So there's a cross-section through one of those dimples, and so it's got very specific layering of chitin in air, but it's also got this very specific curvature to it. And that's how it gets the color. So what happens is that white light hits the bottom of that dimple, the spacing is right to get constructive interference of yellow light. But when white light hits the edge of the dimple, of course the spacing is different because of the angle. And what happens is that blue light is what reflects to the other side and it retroreflects up and out. And so what you see is green, but of course really, and these dimples are only about 3 microns across, but what it really is the sum of yellow and blue. And of course then you see that as green. But one of the really interesting aspects of this is because the blue retroreflects, it's polarized. And so they see polarized light. So presumably they don't see themselves as the color of the vegetation. So it's an interesting solution to this problem because if you're really well camouflaged and you don't see each other either. But in this case, they can see each other not as the color of the vegetation. But any vertebrate that doesn't see polarized light, vertebrates don't see polarized light in general, would see them as the color of the vegetation. So they would blend in.
What's their predator?
What's that?
What's their predator?
Birds and lizards.
Okay.
So for the most part, things that can't see polarized light.
But some birds can see polarized light.
What's that?
Some birds can see polarized light.
Can it? How well?
Sorry?
How well?
Um,
I mean, it's great to figure that out because a lot of this is also color is very perceptual, right? And so it's hard to know what the bird truly sees.
The birds of prey, famously.
Yeah.
In fact, it's not big mammals and birds
Right.
have polarized pattern that blends it, helps to blend them in because birds of prey can actually see.
Yeah, but so then presumably they're not birds of prey going after the butterflies, right? So smaller birds. So yeah, but it's an interesting thing. Who really can for sure see polarized light? But one thing that we know is that all insects inherently can all see polarized light, right? Because of the structure of the raptor nerves, they do see it. Yeah.
Are the sizes of these regions dimples random or?
No, they're pretty set. Yeah, there's some variation, but they're pretty reproducible. Yeah. Okay. So the other interesting thing about this particular group of butterflies is that you can actually generate quite a number of colors using this system. So there's 25 species in this group, and they can make anything from a purplish blue all the way to a yellowish green color using this system. So here's one of these examples of blue swallowtail Papilius ulysses. And you can see that it has dimples that create blue, but you might notice that these dimples are far square. Right, and so there's actually no retroreflection; they're just straight edges on them, and they're not curved in the same way. You also get variation within populations for the color. So this is Papilio carna, which lives on a number of islands in Indonesia. In some islands, this area of the wing is blue, and in other areas, it's green. So you can get actually variation within populations in the color. And then you also get an interesting phenomenon known as polyphenism. So adult butterflies can't change their color. But many butterflies have multiple generations during the year. And so this is Papilio maki from Japan, and the summer generation is blue and the spring generation is green. So the same genome, depending on environmental conditions, can generate two different colors. All right. And finally, this example here, this is Papilio numigeni and Papilio pericles. And to my eye, these would appear to be the two most dissimilar in the group. They look very different from each other in terms of pattern. They never see each other because they're on islands 1,000 miles apart. But it turns out they can interbreed in the lab, and you can make hybrids. And so when you make hybrids, you see you have the pattern of this one, but a color that's intermediate between the two. All right, so how do they make these dimples, right? So this was something we thought we could approach because it's a slightly larger structure now, right down to the nanostructure. So we looked at what these guys do when they make scales. So at a certain stage, they look like every other scale that we've ever seen. They have these huge rods of actin in them, okay? But just when they should start breaking down the actin, they do something very odd. So this is the membrane of the scale, all right, at this stage. And now what you're going to see is the actin, but you're going to see it's been reorganized. So it's not in rods anymore, it's in hexagons inside the cell. And this is one big cell that's doing this. But the rods on the bottom of the scale are fine. They're still straight rods. So it's only on the top it's reorganized the actin into hexagons.
This is just a later time
Yep.
in between.
Yeah. So when every other scale that we've studied would be breaking the actin down, it suddenly reorganizes the actin into this hexagonal array.
And this is desynchronized across scales?
So it occurs, as I'll show you in a minute, it occurs in an instant in the scales, right? As far as we can tell, right? So we love to know, okay, so how does it do this? So these dimples then, these hexagons, are then the template for the dimple. So there's actually going to be a basket of actin between those dimples too. And that membrane seems to get pulled down on that, and that seems to be the template for the shape. Okay, so how do you do that? So we can think of some ideas. There's a lot of literature out there on actin and how it forms and all the properties it has. So one possibility is there are proteins like ARP23 that can make 70-degree angles off of the existing actin. So you might think, okay, you could do that, and you could generate a vertex. Another possibility is that you could have proteins or some sort of complex that grabs adjacent rods and pulls them together. In this way, you could get hexagons that way. The other possibility is you could make straight connections between them, and then you could take the whole array and pull it, and that would also generate a hexagonal kind of array. So we thought, well, this will be easy, because all we have to do is just kill a lot of pupae at different time points, and we can see what the intermediates are. So Ryan Null, the graduate student who did all this work, he did that, and basically he found that it went from one thing to the other in an instant, as far as he could tell. That he never saw anything that was convincingly intermediate in the pattern. Yep.
So this transition from rows to hexagons in physical systems appears all the time.
Yes.
And it happens as a function of the aspect ratio of the cell.
So,
Is the
yeah,
aspect
so
ratio changing?
is the aspect ratio.
Meaning the
Which
thickness of the scale compared to the other
way?
dimensions?
No, not at this moment.
Or is it drying out?
No, it's totally wet all the time. So anyway, so what we love to do is actually just watch it live, right? To be able to see how it actually transitions. So the idea would be that we can observe a lot by watching. So, but of course we have a couple problems. One is that the stage where we want to look, the pupae is opaque. So how do we look in and watch what the scales are doing? So Ryan's developed a couple interesting methods in the lab. One is that we have actually figured out, at least for certain stages, how to grow the wing in a dish. So these are wings removed from pupae, and then you can see them developing, and you can see the colors coming in while they sit in a dish. So the problem is that works great if you want to watch pigment colors come in, but it turns out all the structural things are made earlier. If we try to remove the wings at those early stages, they don't survive well. Okay, so then the other technique that we've developed is a technique to make a window into the pupae so that we can see wing development from the moment it's pupated. And so here you're seeing a movie that's 8 days of pupal development. And so from the time it pupated, we can see this hind wing developing just fine. So you're going to see the wing develop. The scales are looking sort of silvery at this point. And then right towards the end of the movie, you'll see all the pigment colors come in, and then the butterfly's fine. Other than it's missing a wing.
How long is that?
That's 8 days. Yeah. And then here's an example where this isn't actually the phalloidin that we normally use to stain the actin, which only works if the cell is dead. Instead, this is LifeAct GFP, so it's looking in a living cell, and we can visualize the actin now. So the idea would be to combine these to be able to visualize the actin in living cells with this window technique to see how it is that you transition from one to another. So that's an ongoing project in the lab, and it's hindered by the fact that you're not really allowed to import, well, at least in California, we couldn't import the butterflies at the stages we wanted into California. Fortunately, in Massachusetts, they seem like, well, everything's going to die if it escapes, so whatever we want to do. So we're looking forward to trying to be able to do that. Another thing that we're interested in is trying to understand how you generate the variation that you see in this group. So you can imagine that you can change both the layering here of the chitin and air, and you can change the geometry. And so we've been focused on the changes that might be due to changes in the geometry. So we've been using various imaging techniques to be able to measure the shape of these dimples in a fast way. And then we can create these kind of 3D reconstructions that show you what the geometry looks like. We can lay over it the image of what light is, what colors are coming from which part of the dimples, so we can get an idea of how the geometry affects that. And then here's an example where it's a different Acolytes species that's not that bluish green color, but more a lemon green color. And And you can see that the dimples actually have this very different geometry where the crosswalls don't meet. So there's no retroreflection in this system. So you just get this green color instead. Finally, I'll end by telling you one more example of another approach we're taking to try to get at structural color. And it's using these butterflies here, which are called buckeye butterflies, which are really common throughout the United States. But they generally lack structural color other than a little bit of blue in the eye spots. Okay? But they are part of a clade, so the buckeye is this butterfly right here. But they're part of a clade which has members in South America, Mexico, and in Africa that definitely have beautiful structural blue and green. And so, it'd be a good group to be able to try to understand how you make structural color. But fortunately for us, there's a woman, Edith Smith in Florida, and she took the buckeye, the North American buckeyes, and every now and then you can collect one that has a little bit of blue or green to it. And what she did is selectively bred them together over a couple of generations. So she was able to get buckeyes that were a beautiful iridescent green color. Okay, and so we were able to get that line from her, and we could ask, well, how is it that these animals have this blue color? And so if you look at their scales, this is one of their scales, it's blue. You flip it over, it's actually the bottom of the scale that's creating the color, and it's a structural color. So if you put it in immersion oil, which has the same refractive index, the color goes away. And if you rip off these ridges, you can see that it's really the lamina, the lower surface of the scale, that definitely is creating the structural color. And the way it does it is in the simplest way possible. So it fits this very simple model that when the chitin is just the right thickness of half the wavelength of what you want to have constructive interference for, that's the color you get. So the lamina of this scale is just the right thickness to give that constructive blue color. Fortunately for us, there was a nice theory paper published from the Stavanger lab that said, well, what if you had lamina of different thicknesses? What colors would you see? And so you can see that blue occurs right around 200 nanometers of thickness of the lamina, and that's what we see in these butterflies. But where did it come from? So they were these brown butterflies, and then suddenly they were blue. But it turns out that those brown butterflies actually are structurally colored. So the scale has brown pigment in it. If you flip it over, the lamina is actually very, very thin, and it reflects a yellowish gold light, or it has constructive interference of a yellowish gold light. And so it turns out that this is not that uncommon in butterflies. That even when they have a pigment color, they actually adjust the lamina to give reinforcement of the color by having that same constructive interference. So what really happened in these butterflies under the selection regime is that what she's selected for is this lamina to get thicker. So it went from about 140 nanometers to about 210 nanometers, and that's what led to the color. Interestingly enough, when we look at these natural occurring species that have blue and green, and again in Mexico and in Africa, that's exactly what they've done. So they've created that color by having a thicker lamina. So what she's selected for in Florida artificially is actually the same thing that evolution had done naturally in this clade of butterflies. So we'd love to be able to see that how it is that you build up this lamina. But I'll show you an interesting thing, that if you're really building this sequentially as you go, you have this prediction that in fact the scale would start gold and then go purple and then blue and then green, right? As it was getting thicker, you should see that projection. And so in fact you see that. So this is that same movie I showed you before of a buckeye wing developing, a few people can see it. But I'm focused on this area which is going to have structural color in it, a bluish green color. And what you're going to see is it's going to go orange and then purple and then through blue and then to a bluish green before the pigment comes in. And so even though we can't see the lamina at that point, we can tell you how thick it is by the color that you're seeing off of it. Okay, but the great thing about having this line of blue butterflies that's just a selected line from the brown butterflies is that we can genetically map the loci that are responsible for this trait. So the idea is that we make F1s, or we've done this already, and the lead is Rachel Fair, grad student in the lab. So she's taken these grandparents, right, made an F1 population of butterflies, and then taken pairs of those and made F2 families. And she's actually been able to make very large F2 full SIP families, one of 501 individuals, another 273. I didn't even know a single butterfly could lay 501 eggs, much as someone could raise them all to the butterfly without losing them. Most of them. So what do the F1s look like? So the F1s look like this. So the F1s actually look more like their brown parents than they do the blue parents. But the F2s are really beautiful. They show all sorts of variation, both in the percentage of the wing that's covered and in the color that they have. So we can group them separately. So we can show you F2s that have the same color but vary the percentage of the wing that's covered by iridescence. Okay, and then we can also look at F2s that have the same percentage of the wing covered by iridescence, but they go from purple all the way through blue to green. Okay. And so using those, then we can then use the QTL analysis to try to find the genes or the segments of the chromosomes that are responsible for these different traits. Okay. And so we've done that. And so if we look at this for percent wing cover, much to our surprise, there are just two giant peaks that explain almost all of the variation in the percentage of the wing that has iridescence on it. Okay, and in this case, we're really lucky because we actually know what that gene is. That gene is a gene called optics. So that gene is responsible for positioning red scales in helicombus butterflies, but it's doing something completely different here in these butterflies, and it's controlling the blue-green color. And in fact, Bob Reed's lab knocked out optics in buckeyes, and now you suddenly see this iridescence color. So somehow optics creates sort of a quantum shift of going almost 2x thickness of the lambda. But it does more than that. It coordinates multiple things. It also gets rid of the brown pigment of the scale. And it actually makes, there's two types of scales, an upper scale and a lower scale in pairs. It makes the lower scale blacker, which you need in order to get the rich hue of the blue. So it coordinates that. What we don't know is what the even bigger peak is. We don't know what that locus is. And so we're homing in on what gene is in under that peak. But maybe more interesting is the QTLs that change the hue, because those are the ones that can very precisely control the thickness of the lambda. And so the results here are not as significant, but they're very promising, that there are a couple of loci that will be able to map and control the hue, that control this pattern in a very fine way. The nice thing is that once we find these genes, there's the protocols for doing knockouts in butterflies are pretty robust with CRISPR-Cas9. This doesn't show any of the genes that control these colors, but pattern instead, and this is a gene called pupae, and these are just some examples of CRISPR mutants that we've made in the lab, and these things. It's just, again, proof for this project, it's just really proof of principle that we can do this and knock out the genes that we want once we find them. So the conclusion from this part then is that, it seems that this transition from these brown scales to the iridescent blue and green scales really has to do with making the lamina much thicker. And that seems to be optics plus some other gene that we hope to discover. But then precisely controlling the thickness to control the hue is some other set of genes which we also hope eventually to be able to narrow down in, but we have good candidates now for the QTL. I'll just end by, without going into any detail, saying the other thing that we've been doing in butterflies is studying transparency in butterflies. So butterflies have also, butterflies and moths, have repeatedly evolved transparency as well. It's generally for camouflage, but not always. And they can be, in fact, wonderfully transparent. And so we're studying how they achieve that transparency. And one of the neat things they do is they also evolve an anti-glare coating so that even when the sun shines on them, they still seem to be transparent. So I'll just thank the people who did all the work. So the initial work on understanding scale development and the active dynamics was done by April Dinwiddie, who is a graduate student at Yale. And she and I collaborated on a lot of that, along with a couple graduate students, or undergraduates in my lab in Berkeley. And then all the work on Palinurus was done by Ryan Knoll, and the work on the buckeyes was done by Rachel Fair. And then a host of other people were involved in different parts of the project. So I'm happy to take questions. Thank you very much.
So in the first example where you had these rows and then you had Christmas trees, do you think that's because you have sort of like periodic deposition of chitin that then builds a tree?
It's a great question, and the shame of it is that no one has any idea. So we don't know if you build that structure inside and then lift it up, or if it's just sequential deposition over time. And no one's ever seen that. And the problem is that, so people will try to do EMs, but they don't know how old the different animals are, and because it's hard to stage them that precisely. And also, you don't know which scale you suddenly cut through. So it's very hard to do. So that's why, again, I think what we really like to do is to watch all of these things live. But we've, of course, set ourselves a real challenge because we're asking to image something that's smaller than a half wavelength of light. So we have to do it by super resolution microscopy, but we want to do it in a living animal. So, but we hope to get there slowly.
This transition from the rods to hexagons? You said it happened instantaneously.
Well, instantaneously in the sense that we can, Ryan's found some animals that he can kind of say have something that's intermediate, which is a little speck of a circle developing sometimes between rods, but that's just sort of
And can you trigger that by chewing the actin gently, by titrating?
We haven't tried anything like that.
Because right now you just dump a lot of whatever.
Oh yeah, then that just depolymerizes the actin.
Yeah.
We had some encouraging results at one point where we thought we could get a shift in the color by depolymerizing the actin at just the right time, but Ryan's not convinced that those results are real. I mean, so obviously what we really, the goal now is really to either with Surac, which is a membrane permeable actin dye, or with a life act to really watch just in a live Palinurus butterfly the transition from rods to hexagons and see what it does.
And the spacing between the rods is
Pretty uniform.
but it changes from the kind of scale in one kind of scale to another kind of scale.
Yeah, so
even in the same.
actually, if you might have noticed that the spacing between the rods on the upper side is twice that of
Yes.
the lower side. So I think the problem is until we see, I think we can hand wave lots of possibilities, and the best thing is to just watch what it does.
So Nipam, in the sort of really fantastic shapes that you gave examples of, do you think it's always the cell shape that precedes, or is it just the deposition of the chitin that has this kind of phenomenal shape?
No, I think for the nanostructures, obviously the scale as a whole is much, much larger than the structure you're talking about, right? So,
But was there cell membrane underlying?
So that we don't know.
Okay.
So like the chitin Christmas tree, do you make a folded membrane and then you put chitin in that, or do you secrete chitin in a way? So the gyroids, people do have this model that what you do is you fold the endoplasmic reticulum and the membrane into a double gyroid, and then you secrete chitin into it. But actually, other people don't buy that model. But that's another one. If we could image it live, we wouldn't need to image the structure. We would just need to image the spectra. We could actually tell you what the intermediate stages were in its production.
I'm presuming there are also things going on beyond the visible spectrum.
Yep. So they also see in the UV. And so they will make nanostructures that create constructive interference of UV light, which we don't see, but they definitely do see. And so that involves an even smaller nanostructure than what we're looking at here in some of the blue and green examples. But they definitely exist.
Would it be possible that any of these structures could be preserved in fossils, say like in amber?
They are. Yeah, they are. So people actually have, there are a couple of published papers, some nice ones in beetles, where they look at the beetle and then they do TEM, and the preservation is good enough that they argue that it was a particular color. So there's one of those talks that I went to, and when I heard the talk, the color was different than when the paper was published, because they decided that, because in the talk it was like red, but then when it was published it was green, and it looked very much like extant beetles. And I think there was an argument that the problem was in the fossilization process, the structures had actually shrunken, and they had to take that into account. But it is possible.
What's optics doing?
So optics is a transcription factor, and in like Heliconius butterflies, it decides, it's the master gene that sort of says be red. Okay, so. And it certainly is expressed in the pre-pattern that matches the red pattern in Heliconius butterflies. In buckeyes, we haven't seen where it's expressed yet. So, supposedly the published literature is it's just expressed in a specialized scale in the margin. But that may not be true. And knocking it out is what gives the iridescence. So presumably it's expressed more broadly. But I think again, it's a transcription factor, but I think the really interesting thing about it is it coordinates multiple events to make them blue, not just the lamp. Yeah.
So the cross strips are also quite periodic, right? Is there a correlation between that and the actin?
The cross ribs?
Yeah.
Yeah, not really. So, and we don't know what sets up the cross ribs. That's another. So what she's pointing out is that there are the long ribs, but between them, and they're structurally important for the solidity of the scale, are cross ribs. And we don't know what sets their position. And sometimes they have structural components on top of them as well.
All right, let's thank Nipam one more time. I also want to thank all the